Good afternoon, everybody. First of all, I want to start off by congratulating my colleagues at ARM for a great 10-year anniversary. A lot achieved, and as always, laying out a fantastic conference for us. This afternoon, I'm going to give you an update on our company, tell you a little bit about what our upcoming milestones are going to be, and happy to meet colleagues later on today to discuss anything you find interesting out of the presentation. Reminder to show you our forward-looking statement. Even as a private company, we would like you to see this. We are a T-cell engineering company that was founded out of the University of Pennsylvania ecosystem. We are private stage, clinically and fully vertically integrated, meaning that we also have our own proprietary manufacturing in-house. And we're focused on developing the next generation of T-cell therapies, be they CARs or TCRs, predominantly for solid tumors and difficult to treat liquid cancers. So our history, founded in 2015 out of a spin-out out of the Penn ecosystem. Nothing unusual about that. Academics spin out companies all the time, but we were the beneficiaries of the first seed funding from the University of Pennsylvania. We subsequently went round and increased the company's financing by doing a successful Series A in the Q1 of 2018, where we raised $135 million. At the time we did the seed raising, we also acquired our own facility in northern Philadelphia, which is our center of R&D now. We licensed over a dozen assets initially from the University of Pennsylvania, and since then have gone on to acquire other assets and license those in from institutions such as UCSF here in California, and you may have seen some news flow about a very interesting target that we licensed in from the Children's Hospital of Philadelphia this week. We're now 52 employees, and we're a clinical stage company. So what makes us unique and different? We were enshrined and founded on the basis of a very, very unique collaboration model, whereby the University of Pennsylvania Cancer Cellular Immunotherapies Group, headed by Professor Carl June, acts as our initial research, discovery, and translational engine, where we work in collaboration with them in terms of creation of packages of data that we then subsequently decide whether to file in through an IND from the company or by utilizing the vehicle of an academic institute like Penn. We probably have one of the most broadest pipelines in the industry right now in the T-cell engineering space, focused pretty exclusively on immuno-oncology, tackling some of the more less trodden cancers, particularly in the solid tumor space. I've already mentioned our expertise from a manufacturing perspective, and I will draw attention to the fact that we also have in-house gene editing capabilities, championing with Penn and the Parker Institute the first in-human dosing of a CRISPR-based product in the US with a program that I will touch on briefly. And we are blessed to have founders who need very little introduction, but have been at the helm of the cell and gene field for well over 3 decades now. So what are we trying to solve for? We believe the central thesis for our company is that if we want to be successful with T-cell based products, or frankly, any product when you're thinking about immuno-oncology and cancer moving forward, how we get into earlier settings is going to be of primary importance. And how are we going to get the radically reduced cost of goods in order to make these businesses scalable and successful? So within that setting, we think we are quite uniquely placed, that one of our key strengths is our understanding of synthetic biology and the ability to not only create constructs in vitro, in vivo, but being able to translate that quickly into the clinic, into an IND setting, because that's where the vast majority of the bottlenecks currently exist, be it in the academic setting or the company setting. I've already mentioned we've been very successful in acquiring a very extensive and broad pipeline within which we are now focusing on our lead programs, but have incubated and are moving forward in a very structured way our preclinical story as well. And the model itself is very unique in that as a company, we don't need to build out internal research and early development, because that is done in partnership with our colleagues at Penn. What we focused on from a build-out internally has obviously been the traditional side of what we would say clinical development and commercial thinking is, but also a very rich and strong bench depth within analytical development, process development, and viral vector know-how within the company. And very importantly, as a thesis of the company very early on, was the idea that we wanted to nail this notion that you have to combine translational medicine with CMC thinking up front, because it just can't be an afterthought. And so that was the basis and the foundation for which the company started. And we've evolved very quickly in terms of thinking about how do we bring the next generation of products through to market. We spend a lot of time scientifically focusing on a number of areas when we look at the microenvironment, but in particular when it comes to solid tumors and difficult to treat liquid cancers like AML, we spend a lot of time on the following areas. So we spend a lot of time as a team thinking about how do we optimize the target. In the solid tumor setting or in any tumor setting, that's of paramount importance. And the ability to be able to affinity tune, whether that be a CAR or a TCR, is of high importance. And also in terms of multivalency, it's going to be very, very important with certain disease states that we actually go in a very bold ambition with multivalency from the start. For example, in cancers like glioblastoma. In terms of actually getting signals within the cell to really supercharge and power on, but also regulate when you need to do that. We have an ability and have demonstrated the fact that we can get into clinic with novel co-stimulation. I'll talk a little bit about that today and why that's important, and also getting these additional boosts that really translate and are very important in solid tumor settings where we need to have products that are going to be persistent and durable, and products that may require reinfusion as well, depending on the tumor type. Overcoming immunosuppression is the holy grail in cancer biology, and we've started to demonstrate that with our CRISPR-based programs, as well as armoring cells with the ability to handle various checkpoints depending on the microenvironment and what it has a propensity to express. And last but not least in that area, it's critically important that we're able to deliver payloads into the tumor microenvironment, especially as we think about autologous products leading into allogeneic products as well, which is an area of interest and research stage discovery for us as well. And last but not least, we're very excited about some of the areas that we're pursuing when it comes to breaching the microenvironment in terms of the stromal surface of the tumor and how we can penetrate that barrier and also introduce not just T-cell therapy, but potentially other combinatorial therapies as well. Our pipeline, as I say, is very extensive and rich. Today I'm going to spend a bit of time talking about our approach in prostate cancer and a little bit about our novel target in terms of solid tumors as it relates to abnormal sugars and abnormal glycosylation. This week we also announced our first deal with the Children's Hospital of Philadelphia for a target called GPC2, which is a great target that we want to pursue in children with neuroblastoma and also medulloblastoma, and also hopefully pursue that in small cell lung cancer, a very aggressive and horrible form of lung cancer, as you know. The other way that we think about our pipeline is the franchises that are now emerging. We're heavily invested now in the CNS space. You saw earlier this year that we announced a deal with the UCSF in terms of a TCR targeting diffuse intrinsic pontine glioma with a TCR that targets histone mismatching. We are also now focused on really accelerating our thinking in glio. The lung franchise is going to be very important for us with the current targets that we have. Pancreatic and prostate are a key feature for us, as well as ovarian cancer. And we are super excited about some of the inroads we can make in terms of myeloma and acute myeloid leukemia. So prostate cancer remains a huge unmet need. These are patients that we are targeting are metastatic castrate resistant, meaning that they have tried all forms of treatment. They've been androgen deprived, they've had medical castration, surgical castration, they've cycled through every androgen deprivation therapy there is there, and pretty much they're coming to the end of the road, but they are still stable with their disease, and you start to see rises in PSA level and a recurrence coming. They may have bony metastases, maybe not have a defined tumor mass because they tend to have a shriveled out disease state at that time point, but this is the kind of patient that we're targeting. And how are we targeting them? So we took a bold decision to go into clinic with an armored CAR as the first offering here. So in the interest of time, I'm not going to actually go through the details of the mechanism of action, but you can see on the left-hand side you have an affinity-tuned CAR that targets a specific membrane antigen called prostate-specific membrane antigen, and we couple that with a dominant negative receptor whereby we're attempting to trap TGF-beta in the environment. And why did we tackle that? So in prostate cancer, as a form of adenocarcinoma, similar to the pancreas, TGF-beta is overly expressed, and I think it's been one of those master regulators that people have underappreciated, but as you've seen of late of the past few years, certainly had a scientific resurgence with a lot of companies approaching the TGF-beta axis for manipulating the microenvironment. So with this program, we're able to also trap the TGF-beta in the hope that it stops the immunosuppression of the cells and the cells power on, and also then treat the cancer. Here is a phase 1 trial that is continuing. There are two actually phase 1 trials now. We have one that I'm showing here, orchestrated at Penn, which is continuing to enroll. Some data has been highlighted from this trial already earlier this year, and we have another trial ongoing where we're looking at both lymphodepleted and non-lymphodepleted settings, and we hope to update with further data there. What I can tell you is that we've seen remarkable cell expansion, even in the absence of lymphodepletion. We've seen biochemical incidences of PSA drops that have been highly significant. We've also seen potent CRS, so we know that we've got a product here that is very, very potent, and we continue to generate data with more data coming out in the first half of next year. TNMUC is our other target that I want to talk a little bit about. This is a target that's going to be looking at various solid tumors where mucopolysaccharides have been implicated over time. Abnormal sugars have been studied for a long time now, but the challenges with abnormal glycosylation is actually finding a way to drug them. So very briefly, on the left-hand side, you see what we mean by healthy glycosylation and normal sugars. So you have the example of a gastric cell where many of you will be familiar with the protective coating of mucin. Mucin is a heavy sugar-coated protein complex. But on the right, you actually see what happens in certain forms of cancer, whereby through some dysregulation of chaperone proteins like cosmic, you can get an abnormal epitope expression of something called TNMUC1. It relates to abnormal hydroxylation of these amino acid chains that leads to this underglycosylation appearance that you see on the right-hand side. And it's this specific epitope of TN and STN that we're targeting with a unique CAR. So on the left-hand side, you will see a schema of what we would call a second-generation CAR, something like Yescarta or Kymriah, more in this example, Kymriah, where you have a 4-1BB co-stimulant, a CD28 CD8 transmembrane component, and an scFv. On the right, you see with TNMUC1, we've actually substituted that and brought in a CD2 co-stimulator. And why is that? We've, through an elegant set of experiments that have been published, have shown that we got better persistence and durability. We're big believers in the 4-1BB approach, but in this clinical setting, we're actually switching to taking forward a CD2 domain. So again, an example of how we move quickly, we iterate in synthetic biology, but getting into the clinic is the paramount importance to us. So this program is now being run in a multicenter setting. We just had a successful IND a couple of months ago. We are now in the active screening phase. We have centers established at Penn. Cedars-Sinai in Los Angeles will be another center, and we will soon announce 2 additional centers that are coming on board. 2 cohorts primarily that we're looking at, a stream on the top for solid tumors, which is a basket trial of this pancreatic, ovarian, and triple-negative breast cohort, and we're also looking at approach to myeloma. TNMUC1 is expressed in a number of these tumors, and so I look forward to hopefully in the future sharing data as it emerges, but an exciting time for the company as we look at a novel approach to glycosylation, how we can target this as an area for cures for cancer. Manufacturing, as I mentioned, has been at the heart of the company from the day it was conceived. We have currently a 46,000 square foot facility in a place called East Norriton, northwest of Philadelphia. It's an organization that we've established now with, as I mentioned, very strong bench strength depth in analytical development, process development, and viral vector. It can be expanded to 200,000 square foot, and it's our hope that by 2020, our GMP manufacturing will all occur in-house, as well as utilizing the manufacturing that we have unique access to at Penn. And we would like to then instigate our next generation of manufacturing out of that facility there. We're already looking ahead at other areas and highlights for the forthcoming year that I think are going to be important for the field, but also for ourselves. The idea that you cannot get away without having active comparators is a big, big challenge for us in the field, especially as you're looking at cancers in which randomized control trials are extremely challenging. So how we embed synthetic comparator arms, that's a priority for companies like us, and we'll have some deal flow news around that. Novel pricing and reimbursement, I think those of you who were at the ARM panel this morning know the challenges and having the benefit of seeing what happened with the Kymriah journey from my former life, and also where the field is moving. It's going to be vitally important we focus on that. It's a whole new world, a whole new business model. That's why many of you are here today, as you think about how the businesses are going to be scaled in the future, and of course, chain of identity and how we really wire. We've done it for hematological malignancies. How are we really going to do this efficiently for solid tumors is the next piece of the puzzle that we need to solve for. So in conclusion, thank you for making the time to listen to our story today. I think as a company, we've made tremendous progress since our inception a couple of years ago. We have a rich bench strength of talent now. We have an amazing pipeline, and we're in the clinic. and we look forward to sharing more data with you in the 2020 horizon. So thanks for your time.